the difference between cyclic and non-cyclic photophosphorylation, and then we'll talk about the next part, that is light independent part. Okay. Cyclic and non-cyclic differences. First is what? Which system, pigment system is involved? Only PS1. Whereas non-cyclic photophosphorylation shows both pigment system 1 and pigment system 2. Then electron moves in a closed circle, that is in a cyclic manner. From whichever the chlorophyll molecule it is released, that electron is released, it comes back to the same chlorophyll molecule reaction center. But here it is not of closed circle type, but and then water is the ultimate source of electrons here, because splitting of water takes place, as I told you. Then reduced NADPH2 is not formed, and assimilation of CO2 is slowed down in case of cyclic. Have you observed any NADPH2 formed during cyclic? No. But here you have observed whatever the hydrogen ions are there, they are accepted by NADP to form NADPH2. So this is evolved, and there is assimilation of CO2, carbon dioxide. Then during that process, oxygen is not evolved in cyclic way, whereas oxygen is produced during the non-cyclic way, because splitting of water takes place. Okay. Then this system is found dominantly in photosynthetic bacteria, whereas this system is dominant in all type of green plants. All green plants show which way of photophosphorylation? Non-cyclic. Whereas photosynthetic bacteria, they show cyclic way of photophosphorylation. Okay. Then this is what, these are the main important differences between cyclic and non-cyclic type of photophosphorylation. Now, this, what does it mean by chemoosmosis? That part I will not right now tell you, because first, in continuation with this light dependent reaction, the next is what? Light independent reaction. Okay. So light independent reaction means what? It doesn't require light directly. So in absence of light, this reaction takes place. Okay. So photosynthesis is showing how many phases? 2 phases. One is light dependent and the second is light independent. So that light independent reaction is also called as dark reaction, because it doesn't require light directly. Okay. Then this is also known as Blackman's reaction also, and this is well known by Kelvin cycle, and which occurs in stroma of chloroplast. This is one more difference you need to remember. Both the light reactions took place where they take place? In grana, because thylakoid membrane is required, right? Which is capturing the photons, that is light energy. Whereas this light independent means dark reaction takes place in which part of the chloroplast? Stroma. It requires CO2, because third step is what? Now first 2 steps involved in light dependent. One is capturing of light and then converting it into chemical form, that is ATP, not chemical, ATP and NADPH. It is stored, that energy is formed. Now this energy is utilized where it requires CO2 to form glucose, that is food material, carbohydrates. So it requires ATP and NADPH, which is produced in light reaction, and that forms glucose, that is sugar, from CO2 and hydrogen. So these are the main points which are seen in this Kelvin cycle. This light independent reaction that shows 3 to 4 phases, that I will explain later. But these plants which show this cycle, they are called as C3 plants. And why this cycle is called as C3 cycle? Because the first stable compound which is produced contains 3 carbon compound. So that's why this is called as C3 reaction or C3 pathway, not cycle. Kelvin cycle and C3 pathway is one and the same. Why pathway? It is showing that in a sequential way, that transfer of the conversion of one compound to another compound. At the end, there is synthesis of glucose molecule, right? So this is C3 pathway, also known as C3 pathway, and 80% of the plants on earth show such type of Calvin cycle, that is C3 pathway. Then where it takes place, purposely I have drawn again, I have taken this diagram. This is what granum, where light reactions takes place, whereas this is stroma, where now which we are going to discuss, that is the dark reaction or light independent reaction or Blackman's pathway, or which is the most commonly known Calvin cycle. So one and the same, C3 pathway, Calvin cycle, dark reaction, light independent reaction, and Blackman's pathway, C3 pathway. All are same. Any name can be asked. Okay. So little information you should know about Kelvin. Kelvin is the name of the scientist. That is Melvin Kelvin is the scientist who first of all explained this pathway. And so this is the second stage of photosynthesis, as you know. That is the second dark phase of photosynthesis. And in 1961 Time magazine, he was also named as Mr. Photosynthesis, because this pathway is very important. He gave this pathway, and that's why well known as Kelvin pathway, Melvin Kelvin. He first of all explained this pathway, sometimes referred to as light independent reaction or dark reaction, and because the Kelvin cycle doesn't require light directly. Now these are the steps which are taking place in dark reaction. Now be very careful and try to understand. Once you understand, it is not difficult, and I will revise again at the end. Now what you remember in dark reaction, there are three main steps. Okay. First is what? Fixation of carbon. This is also called as carboxylation phase. Second phase is called as reduction. Third phase is divided into two parts. One is the synthetic part, and the second part, that is, suppose A is considered synthetic part, third reaction. Third B will be considered as regeneration stage. So how it takes place, I will show you with the help of this diagram. See, you know, when this reaction is taking place in the stroma, okay, stroma of what? Chloroplast. Chloroplast, where it is present? Leaves. So what? How the gases enter the leaves? How gases enter the leaves? Through which part? Openings are present in on the leaves, na? Upper epidermis, lower epidermis. What are these openings called as? Stomata, isn't it? So through stomata, there is entry of the gases like CO2. What is required now for the process, this dark reaction photosynthesis? CO2 is required, isn't it? So entry of CO2 takes place. But in the stroma, the substance called RUBP, ribulose biphosphate, is present, which when CO2 is entered, it reacts with this RUBP. RUBP is ribulose biphosphate. Now, can you see the number 1, 2, 3, 4, 5? How many carbon atoms? These shown by red color, these are the carbon atoms. Okay, so this is 5 carbon compound. RUBP is what? 5 carbon compound. Biphosphate, bi means what? 2. This is one phosphate and this is another phosphate. Understood? So ribulose biphosphate is present in the stroma of the chloroplast. When CO2 enters through stomata from the atmosphere, this CO2 is accepted by RUBP, that is 5 carbon compound. Then with the addition of this carbon in the form of CO2, how it will become 6 carbon compound. Okay, so there is formation of 6 carbon compound. See, from the atmosphere, this one more carbon is added, right? Okay, now, but what happens when it reacts with this 5 carbon compound? 6 carbon compound is produced, but that is very unstable. That 6 carbon compound, whichever is produced, that is very unstable. It immediately gets split into 2 molecules of 3 carbon compound. Are you getting my point? When 5 carbon compound RUBP receives carbon in the form of CO2 with the entry of CO2 in the stroma from the atmosphere. 6 carbon compound is produced, which is very unstable. Immediately that 6 carbon compound gets splitted into 2 molecules of each containing how many carbon? 3 carbon compound. So this is the 3 carbon compound called as phosphoglycerate. Can you see now 3 carbons? Phosphoglycerate, the position and how many? From a single molecule of RUBP, 2 molecules. So if 3 molecules of RUBP are there, how many molecules will be produced? 3 carbon compound, how many molecules will be produced? If 6 RUBP are there, how many molecules of these 3 carbon compounds will be produced? 12. Is it okay? Now, if there are here suppose 6 molecules of RUBP, then with the entry of then again 6 molecules of CO2 will be required. And then how many molecules? 12 molecules of 3 carbon compound will be produced. Is it understood? Clear? So here 3 carbon compound called phosphoglycerate or phosphoglyceric acid also it can be said is produced in presence of enzyme carboxylase, that is also called as rubisco, that is ribulose biphosphate carboxylase enzyme. Carboxylase means what? Addition of carbon. Here in the form of CO2 there is addition, isn't it? So carboxylation reaction takes place. So this step is also called as carboxylation stage phase, or this is also called as fixation phase. Fixation of what? Carbon, that is in the form of CO2. So this stage, this phase is called as fixation of carbon dioxide, that is called as carboxylation, where addition of carbon in the form of CO2 takes place. So this is what carboxylation phase. Up to this, is it clear? Now what happens? This 3 carbon compound, that is 3 phosphoglycerate, gets converted into 1,3-PGA. 1,3-PGA, 1,3-phosphoglyceric acid or 1,3-phosphoglycerate, where this ATP gets converted into ADP. Now ATP is produced in an light reaction. You understand? You remember light reaction, it produces ATP. Now that ATP is utilized by 6, because here 6 molecules are shown. So that's why here 6 ATP for 6 carbons, 6 for 3 carbons, 6 molecules, 3 carbon compounds. So it is doubled. So here 6 ATP will be utilized to form ADP and IP. So this is what reduction of ATP to ADP. And at the same time 1,3-DPGA gets converted into PGAL, that is phosphoglyceraldehyde. So that is the another compound with again the reduction of NADPH into NADP. NADPH is also produced in first stage of photosynthesis, that is light reaction. So ATP and NADPH, which are produced during light reaction, here they are utilized and they get reduced to ADP and NADP, right? That's why this phase is called as reduction phase. Are you understanding? You understand what I am saying? Try to understand. I'll revise again once again, but try to understand. Okay, are you getting my point? Whatever I'm saying. First, how it is carboxylation or fixation of CO2 I explained. Then with this PGA, phosphoglycerate is formed, which is 3 carbon compound, right? So this is the first stable 3 carbon compound. So this is also called as C3 pathway. Are you getting? Which is the first stable compound in this pathway? PGA, phosphoglycerate you can say, or phosphoglyceric acid also you can say. Okay, so this is the first stable and that is 3 carbon compound. So this is called as C3 pathway. Now after this PGA phosphoglycerate gets reduced to PGAL with 2 continuous consecutive phases where ATP gets reduced to ADP and NADPH gets reduced to NADP. which ATP and NADPH are produced in light reaction, now they are utilized in the dark reaction. Are you getting? Now, out of, excuse me, is this connected? Trace load. Plugin PC. Okay, we'll see now. What I was talking? Second phase, is it clear? That is called as reduction phase, where this phosphoglycerate gets converted into 1,3-diphosphoglycerate first and then to phosphoglyceraldehyde. Here, then again up to this, you can see the 3-carbon compound. Okay, here as the number is taken here 3, so here it gets converted into 2 molecules of each, so total is shown here as 6. Whatever the number, accordingly as those many turns takes place or those many molecules of CO2 are utilized. Okay, so then here is the, this is why called as reduction, because ATP gets reduced to ADP and NADPH gets reduced to NADP. Now, whatever, out of, now suppose here are the 6 molecules of PGA as per this diagram. Okay, now what happens? One molecule of PGA, sorry, not PGA, PGAL is produced. That is the reduced compound, reduced form. PGL, that is phosphoglyceraldehyde. It again gets converted into DHAP, that is dihydroxyacetone phosphate, which are the isomers of it. They get converted, interconvertible molecules. They are interconvertible molecules. It is not shown here, but just remember, I will show in one more diagram. So these, then PGAL gets converted, that PGL, that means one molecule of PGL is utilized to form glucose, where it enters C3, that is Kelvin cycle C3 pathway. That means here, sorry, this is here one molecule of of this reduced compound is utilized to form glucose molecule. So here is the synthesis of glucose. So that is what called as synthetase phase or synthesis phase. What is synthesizing here? What is synthesizing? I told you just now. What is synthesizing? Glucose. Obviously, that is the main part of what this cycle. At the end of the photosynthesis reaction, what is produced? Carbohydrates, food in the form of glucose. So out of, if there are the 6 molecules, then one molecule is used for the synthesis of glucose, whereas remaining 5 molecules are used in the regeneration of RUBP. And that's why this phase is called as regeneration phase. What? Why? Re is what? Again. Generation of what? RUBP. Again, RUBP is regenerated. That is a 5-carbon compound with a series of reactions. This may get converted into sometimes 4-carbon compound, 5-carbon compound, 7-carbon compound, and then at the end, RUBP, which is the 5-carbon compound, is produced. So this is what regeneration of RUBP from this 3-carbon compound, that is phosphoglyceraldehyde. And that is why this is called as regeneration phase. That's why I told you that this step, after the second step, it is divided into 2 parts. One is called as synthetic phase, where there is the synthesis of glucose molecule, and the second, that remaining 5 molecules, they are utilized in the regeneration of RUBP. And during that process, ATP again is utilized to form ADP and IUP. So what happens in light reaction? ATP and NADPH are synthesized, whereas in dark reaction they are utilized. Are you getting my point? Now with one more cycle, this diagram, again I will show you. If, see, this is what RUBP. 5 carbon compound, can you see? These are the 5 carbon compound, RUBP, then gets converted. This is what? RUBP. There is interconversion, conversion of RUBP into 1,5 biphosphate also. That is also 5 carbon compound. Now, how many molecules are used here? 6 molecules. When atmospheric CO2 enters this 5 carbon compound RUBP, then first of all gets converted into here unstable compound, 6 carbon compound. With the addition of this carbon into 5 carbon compound, it becomes 6 carbon compound, but immediately it splits into 2 molecules of how many carbon compounds? 3 carbon compounds. And so if there are 6 molecules of RUBP, how many molecules will be produced? 12 molecules of 3-PGA, phosphoglycerate or phosphoglyceric acid, you can say. And this is what carbon fixation. Carbon is fixed. That is addition of carbon. That is carboxylation. So fixation of CO2 or carboxylation is one and the same. Either you can call as carbon fixation or you can call this step as carboxylation. Understood? Then this PGA is reduced to PGAL with the utilization of ATP and NADPH, which are produced during light reaction. So that's why these are shown like this. As there are 12 molecules, how many ATP are required? 12 ATP. Here, isn't it? These 6 molecules, after splitting, got converted into 3 carbon compounds. That means 12 molecules are produced. So both not understood? See, this is RUBP 5 carbon compound. Is it clear? When atmospheric CO2 enters, then it becomes 6 carbon compound. But this is very unstable. Here it is not shown. So this 6 carbon compound immediately splits into 2 molecules of 3 carbon compound. And that's why this 3 carbon compound is what? Phosphoglycerate or phosphoglyceric acid. Up to this, is it clear? But here, either 6 tons of entry of CO2 will take place, or 6 molecules of RUBP will be used, and at the same time 6 molecules of CO2 are required. And that's why after splitting 6 molecules into 2 molecules of 3 carbon compound, what will be the number? 12. When there are 6 molecules of 6 carbon compound, but splitted now into how many? 3 carbon compound. 1 into 2 means 6 into 12. Is it okay now? Is it clear? And this is what? Carboxylation. And then this is the second stage, is what called as? What is the second stage called? Reduction. This phosphoglycerate, by using ATP and NADPH2, gets reduced. Okay. And where it gets reduced, that is also the 3 carbon compound PGAL. Here the name is not given, but that is through consecutive 2 reactions, PGAL is produced, and this phosphoglyceraldehyde and DHAP, they are interconvertible molecules which gives rise to synthesis of glucose. Out of 12 now, so both here up to this, is it clear? There are 12 molecules. Now out of 12, 2 molecules are used for the synthesis of glucose, whereas remaining 10 are used for the regeneration of RUBP. So that's why here it is shown. Now these 2 molecules are used in the synthesis of glucose, whereas remaining 10 are used in the regeneration of RUBP. Is it clear? Yes or no? Are you understanding? No? Is it clear? Little bit, yes, okay. Now see, this is the diagram which is shown in your textbook. Again, much more clear, I will show you what is this synthetic phase. Okay, see, 6M is written, that means what? 6 molecules. Everybody, I know this is little different and little bit complicated, but doesn't matter. Again and again when you read, you will understand. I am trying to make it simpler, but still might happen that you might not understand, you may not understand, but it is okay. When you revise this part now, that will be fine. Then you will be understanding. What you will remember, simpler form, I'll tell you now. What you will remember in this dark reaction or C3 pathway or Kelvin cycle or Blackman's reactions, all one and the same, or light independent reaction. What will you remember? You remember the 4 steps. Means third is divided into 2, so that's why 4 steps I'm telling. First is what? Now you tell me. First is what? Carboxylation, right? So carboxylation is addition of carbon in the form of CO2. CO2 is entered through stomata, where in the leaves, what the leaves contain. So chloroplast, which part? Stroma. There, what is present in the stroma? This RUBP is present in the stroma. What is that RUBP? Ribulose biphosphate, that is 5 carbon compound. Okay? Is it clear up to this? Now let it be about this, how many molecules? How many molecules, that is okay. If only one molecule of RUBP is considered, right now you just consider only one molecule of RUBP is there. So how many molecules? Only one molecule of CO2 is required. One molecule of RUBP will receive only one molecule of CO2. If 6 RUBP, then how many 6 molecules of CO2? Or 6 tons of CO2. Either or. Either 6 molecules of CO2 at the same time, or 6 tons of CO2 for the synthesis of one glucose molecule. So this is the carboxylation stage. Is it okay now? First step, carboxylation, where addition of carbon in the form of CO2 takes place, where unstable compound, that is of 6 carbon compound, but immediately it gets splitted into 2 molecules of 3 carbon compound. Okay, so that phosphoglycerate is produced, that gets converted into 1,3-diphosphoglycerate and then to phosphoglyceraldehyde with the reduction of ATP to ADP and NADPH to NADP, because these 2 molecules are getting reduced. Okay, and that's why this step is called as what? Reduction state. Like carboxylation, you understood addition of carbon. Here reduction of ATP and NADPH is taking place. That's why this is called reduction state. Now the third step is divided into 2 parts. Suppose this is A and this is B, you consider. So how it is divided? Half part is considered to be synthetic phase and half part is called as a regeneration phase. Synthetic phase, why? Because synthesis of what? Glucose is taking place, isn't it? So here from 12 molecules, 2 molecules are utilized in the synthesis of what? Glucose through series of reaction. So phosphoglyceraldehyde gets converted into DHAP, that means dihydroxyacetone phosphate, and this is again getting converted, that these are interconvertible molecules. Okay, that is also 3 carbon compound. So that is getting converted into fructose 1,6-diphosphate, and then fructose 1,6-diphosphate again gets converted into fructose 6-phosphate with the formation of 1 molecule of ATP, and then fructose 6-phosphate gets converted into glucose 6-phosphate. This is the isomer of this fructose 6-phosphate, and glucose 6-phosphates are just isomers of each other. So this reaction is called as isomerization reaction. And then at the end, glucose 6-phosphate gets converted into glucose, and 1 more molecule of ATP is again generated. Okay, and this entire stage is considered to be synthetic phase, where out of 12 molecules, how many molecules are used for the synthesis of 1 molecule of glucose? 2 molecules, and remaining 10 molecules are used to regenerate what? First compound is what? First carbon acceptor is what? RUBP. So RUBP is the first carbon acceptor or CO2 acceptor in this reaction. Okay, through series of reactions, then these 10 molecules, which are 3 carbon compound, may get converted into 5 carbon compound like xylose 5 phosphate, 4 carbon compound erythrose 4 phosphate, or pseudoheptulase. Pseudoheptulase is what? That is 7 carbon compound, and at the end again it gets converted into RUBP. Then through then RUMP, that is monophosphate. RUBP is what? Biphosphate. That is what these are the molecules. And then from RUMP again RUBP is produced. So through series of reaction again there is regeneration of what? Regeneration of what? What is this? Produced. Regenerated, generated again, synthesized again, produced again. That is what? RUBP. Isn't it? So that's why this is called as regeneration stage. Is it okay? Understood? And this RUBP again enters this cyclic way. That is the same pathway. That is C3 pathway. Why C3 pathway? Because the first stable compound is what? PGA. Phosphoglycerate or phosphoglyceric acid is the first stable 3 carbon compound. So this is also called as C3 pathway. Is it clear? Yes. Good. Now, these are, if only cycle is asked, you will be drawing this diagram, which is there in your textbook. But if the reactions are asked, these are the 4 steps. You can say 3 steps. First is carbon fixation, where you can see RUBP, entry of CO2, or directly you can write down plus CO2, like this RUBP plus CO2. What it gives rise to first, that is unstable compound, and then it gets converted into phosphoglycerate. 3-phosphoglycerate. Okay, so this is what the first step, that is carboxylation or carbon fixation. Then the second reaction is what? Reduction, where you can write down what is produced now? 3-PGA plus, what you can write? 3-PGA plus ATP plus NADPH, and what it is giving rise to? PGAL plus, what this is converting into ATP into ADP plus IP, and here NADPH is getting converted into NADP plus H, right? So this is what? Second reaction. And third is what? Third reaction is what? Regeneration, where you can write down in the regeneration that PGAL giving rise to 2 molecules glucose, that is synthesis phase. Synthetic phase or synthesis of glucose, and remaining molecules, that is regeneration of RUBP. That means that last whatever you write, that is giving rise to again 5 carbon compound, that is RUBP. So this RUBP molecule go through a complex series of reaction, form 3 carbon compound, and this continues. This cycle, once again there is regeneration of RUBP, this cycle is continued with the addition of CO2 again. Now I want to show you how these light and dark reactions are interdependent. The question only can be asked, how dark reaction and light reactions are dependent on each other. Now you only tell me, without looking at this diagram also you can tell me. I explained while explaining this dark reaction. You tell me what is utilized in light reaction and what is produced. Same, what is utilized in dark reaction and what is produced. Okay, I'll tell you with the help of this diagram. This is light reaction, where it is taking place? Grana of chloroplast, right? Now, what is taken? Light is taken. The thylakoid membranes, what they are accepting? Light energy, that reaction centers, right? Trapping of the light energy and stored in the form of what? What is produced? ATP and through non-cyclic NADPH, right? And what is released? Oxygen is released. Okay, now dark reaction, what it is taking from the atmosphere? CO2. And what is produced? Glucose. And but what is utilized? ATP and NADPH. produced by light reaction, they are utilized for the process of dark reaction, but at the same time, what is released? NADP and ADP as produced, which are again utilized by light reaction, isn't it? So in this way, light reaction and dark reactions are interdependent. They are considered to be interdependent. They are dependent on each other. Is it clear? Have you understood? Any other doubt? Okay, we will discuss this first and then we'll talk about the doubts, your doubts. Okay. So first, this is the difference. Hillman reaction is what? Or Hill reaction? That is a part of light reaction. And this is called as Blackman reaction, because that is the name of the scientist also. Okay. And this is also called as what? Dark reaction. Whereas this is called as light reaction, which requires light, where it takes place in absence of light, right? Then photolysis of water takes place, and what is generated? ATP and NADPH. What is utilized here? CO2 is accepted and that gets reduced to form what? Hexose sugar, glucose too. Glucose is produced, isn't it? During this part of photosynthesis, that is the light independent part, isn't it? Which is called as dark reaction. So light reaction is called as photochemical, whereas dark reaction is called as biochemical reaction. These are the differences. Got it? Understood? Any difficulty? Any doubt? Have you understood? Everybody? If you have doubt, you can ask me. Otherwise, we will discuss a small part, very small part. Is it okay? Have you understood? Okay. So, yes.
After the synthesis of glucose, when ATP molecules are released, so where does this fall?
Which ATP? In
wait
a minute. Just tell me the page number. I will see. One. You are talking about synthesis phase?
Yes.
Here ATP, two molecules of ATP are used, produced, no?
Yes.
Yes, they are produced, they are stored, and again they are used.
For the regeneration of amino
No. For regeneration, 10 molecules of this PGAL are used, and 2 molecules are used for the synthesis of glucose. And that amount of energy is stored in the form of whatever ATP is stored in the form of, the energy is stored in the form of ATP only. So, where the ATP is stored, which part of the cell? Correct. Because this is the usable form of energy. Understood? Any other doubt? Anybody, any doubt? Otherwise, I will take a small part of this, that is the photorespiration. Very simple. Do you know the respiration, right? What is taken, what is done? Simple form in respiration, what is done? Inhalation of oxygen and release of CO2. What in all type of aerobes? Plants also respire. Plants also respire. But that is called, in presence of light, sunlight, it is taking place, so photorespiration. So what is then the photosynthesis is? Photosynthesis is little different than the photorespiration. Now we have learned photosynthesis part, light dependent, light independent, okay, where atmospheric CO2 is utilized and oxygen is released. But at the same time, what happens? Atmospheric oxygen, it's not fixed, right? Only CO2 will enter and O2 will not enter. Is it like that? Through the pores, all the gaseous exchanges take place. But what happens? Photorespiration in presence of light, most of the plants, especially C3 plants, what they do, they respire. How they respire? means what? CO at the same time, along with the CO2 fixation, oxygen is also fixed, because what is the first carbon acceptor, CO2 acceptor in Kelvin cycle or C3 pathway? RuBp, correct? So RuBp is the substance which have affinity with both, less affinity with CO2 and little bit more affinity with oxygen. So not only carboxylation but oxidation, addition of oxygen, there is entry of oxygen also taking place for respiration. What is done? Oxygen is taken in and CO2 is released. So like carboxylase enzyme, oxygenase enzyme also is present over there. Okay, and so at the same time, though CO2 is fixed for the process of photosynthesis, but at the same time, as this is having the affinity, RuBp is having the affinity with oxygen also, it takes oxygen also to some extent, and then what happens? CO2 is released through respiration, but that is released out in the atmosphere. So that is not utilized by the plants for the process of photosynthesis. So this is what loss of CO2. And what is the this is what photorespiration. Have you understood photorespiration? I will tell you with the help of these slides now. So generally where it occurs in hot, dry, bright days, stomata are closed, fixation of oxygen instead of CO2. When it takes place, then only photorespiration takes place. Then produces 2 carbon compounds instead of 3 carbon compound. Now last time, what is the first stable compound we discussed just now? PGA, 3 carbon compound. And it doesn't produce any sugar molecule, neither it produces ATP. When photorespiration takes place. When photorespiration is taking place, these are the criteria when it takes place. Okay. Then because of this photorespiration, plants have spatial adaptations to limit the effect of photorespiration. And these photo adaptations, that means the photorespiration, these plants which show the spatial adaptations, these are called as C4 plants and CAM, that means CAM plants. Now what is C4 plants and what are CAM plants and what is C4 pathway, that we will discuss tomorrow. C3 pathway is clear. We have revised the entire part of yesterday, as well as we have discussed today only light independent, that is the dark reaction part of photosynthesis. Now, why we are studying C4 plants or C4 pathway or CAM plants? I gave you the reason also. That is photorespiration. Okay. Now, the reason behind this, some of the plants, when they are doing the photosynthesis process is going on, some amount of CO2 is released out in the atmosphere, which is not used up by the plant. And so some plants, they have adapted themselves, which are generally growing in dry and high temperature conditions. They have adapted themselves, they have changed their internal structure, that is anatomy, and they show the alternative pathway, that is called C4 pathway, which we will discuss tomorrow. I will stop my presentation as well as I will stop my recording.
Open your textbook.